Rizal Hariadi. So Rizal is originally from Indonesia. And after Indonesia, in Indonesia, he competed on their physics Olympiad team. And after that, he came to University of Washington State, where he did his undergrad degree, and then he did a PhD at Caltech. After Caltech, he did postdocs at University of Michigan and then also at Harvard. He joined us here at ASU back in 2016. And since he's come to ASU, he works with a very interdisciplinary team, which he'll tell us some about today. And he has won a couple of awards, the Arizona Biomedical Research Commission New Investigator Award, as well as the NIH Director's New Innovator Award. And I also wanted to mention going forwards in the colloquium series, one of his PhD advisors, tutor, he wasn't your official advisor, was he, Eric?
I had two PhD advisors, one of whom
yes.
will be here.
One of whom is coming later in the semester. So if you want to hear even more about this material later in the semester, there will be related future colloquia. Okay, so Rizal is going to tell us today about probing biomolecular fluctuations under tension along multi-axial entropic spring tweezers, with multi-axial entropic spring tweezers along ring-shaped origami. And I am assuming he will tell us what all of that means. So here you go.
Thank you. Whoa.
Okay. Do you want to switch him the other way?
Good.
This is good. Everyone can see? Okay, let's go.
Thank you, Cindy, for the introduction. So I'm very excited today to share with you the latest development of an experiment that we've been doing in the lab. So we're developing an innovative tool for studying biomolecular fluctuation and processes under multi-axial tensions. So instead of using magnetic fields or electric fields, we use entropy. That way we can shrink the tool that we use. So typical tools for this study is like the size of a very small lab. We shrink the tool to molecular scale. So my name is Rizal. Again, I'm running Bionics Lab. It stands for Biomolecular Nanosystems of Ever-Increasing Complexity and Scale or Size Laboratory. We are driven by two scientific questions. One is, can we advance our understanding of emergent behaviors of complex systems, such as self-assembling systems comprises DNA subunits or perhaps collection of molecular machines. And the second question is, can we employ these nanostructures to investigate the intricacies of biological physics on the molecular scale? So we study across scales from molecular scale here to centimeter scale. So on the centimeter scale, I will show you at the end of the presentation data that use DNA origami nanoarrays. On the other extreme, we study membrane biophysics using synthetic approach. We built membrane associated or the analog of membrane receptors. And that led to a formation of a spin-off company on ASU campus, ASU downtown campus with Haoyan. But for today, I will only talk about protein or biomolecular fluctuation under multi-axial tension. This is the brainchild of a very talented physics graduate student in the lab, Bima. He is in the room. This work is supported by NIH Director's New Innovator Award, and Bima is supported by American Heart Association Fellowship. So I hope you don't mind if I share something personal. This is a snapshot of the website of Eric Winfrey this morning. I first read this website when I was an undergrad many years ago, and it sticks with me. This is a list of questions by Eric Winfrey's a PhD advisor, John Hopfield. He is renowned for the kinetic proofreading theory, Hopfield network, and he notably served as the president of American Physics Society in 2006, I believe. So today I will start with, or I will put the, I will frame this lecture in the context of the first question. Basically, how can life emerge from weak interactions between inert molecules? So this is one of my favorite questions. I ask these questions to first year graduate students and my colleagues. If you ask students and colleagues in the department, they may invite you to read this book here. It is written by Erwin, not really written. This is actually a compilation of his lectures. So the story was Erwin Schrödinger was, of course, an Austrian physicist. He came up with postulates and Schrödinger equations. But he also studied Dublin Institute for Advanced Studies. And during his tenure, he delivered many thought-provoking lectures that people compile into books, okay? So I don't have time to go through the whole book, of course. But if you ask my colleagues again, for example, Rob Phillips, or even if you go to Wikipedia, I was surprised this morning that the Wikipedia page highlight the same sentence. People often think that this sentence in the book summarize the whole book, which is, so let me read it for you. How can the events in space and time, which take place within the spatial boundary of a living organism, be accounted for by physics and chemistry? And some people even shrink it or summarize it even further, saying that the summary of the book is those two words, accounted for by physics and chemistry. Okay. So what do we mean by that? Apologize if the screen is not clear. We can take less. What do we mean? What do physicists mean by accounting by physics? So we can take a lesson from hundreds, like many years of history of understanding spectral lines or white light. Okay, if you this spectral lines the spectrum may look simple, but if you have impure, not like a mixture of gases, the lines will be very complex. It took scientists and physicists and mathematicians a long time to understand, to predict the positions of the wavelength. In 1885, Balmer, who was a high school math teacher, he was properly trained in math, came up with Balmer formula, which I didn't write. This is a Rydberg formula that probably you're more familiar with, that could predict the position of those wavelengths. So that's accounting using math without full understanding. People then use the formula and codify it in physics or in math and physics, and then led to the development of Bohr atom, and that thing led to the development of Schrödinger equation. And now we routinely ask our first-year graduate student, fourth-year undergraduate students to use Schrödinger equation in spherical coordinates, and they should arrive at Rydberg formula. They can even express the Rydberg constant in terms of fundamental constant of nature. Okay, so that's the standard of accounting by physics. Okay, so can we do the same accounting in using biomolecular systems? So this is data from Eduardo Perozo and Boris Martinek. They visited ASU before the pandemic, and I had a pleasant discussion with him. So what they did is they study mechanosensitive ion channels, but briefly, ion channels are membrane-associated. They let ions rush in or out, and when ions goes in or go out, the current change, right? And then it can also close and block the flow of current. So the data on the top left is a single ion channel recording of opening and closing. When the current is high, the ion channels are open. When the current is low, the ion channel are closed, okay? So from this telegraph type of signal, Eduardo Perozo could calculate the probability of open, basically the fraction of high current states. And he came up with empirical mathematical relation to curve data that fits data really well. But since this is physics colloquium, we can do better than that. We can explain the data in a more satisfying way, and the math is very familiar to all of us. This is the same math for two-state systems. For example, spin 1/2 particle in magnetic field. So we have two states, open and close. with their associated energy, delta G closed and delta G open. The degeneracy are 1. And then we just use Boltzmann distribution, which basically the probability of state I, a given state, is proportional to the E to the minus the change of delta G, the change of energy, free energy, divided by KT. And to normalize it, we divide everything by a partition function. So we could derive the probability of open state using simple statistical mechanics. And we can then compare our formula. And what I argue is that it is more satisfying to invoke physics to curve fitting. So right now we know that this alpha fitting parameter has 1/KT component to it. And we get the negative sign for free because due to partition function and Boltzmann distribution. And we know what this P1/2. So if we put X is equal to P1/2, then the probability open is 1/2. Then basically the P1/2 is the pressure at which the delta G of open state is equal to delta G of the closed state, okay? So, and then what next, right? So we can then return to studying or re-examining the history of studying hydrogen atoms. So to further test or expand our theory, scientists expose or subject hydrogen atoms to, let's say, magnetic field that led to the discovery of Zeeman effect, or electric field that led to the discovery of Stark effect. Right? In biology, what many single molecule biophysicists do, we expose it to mechanical forces because mechanical forces are important in biology. Okay. So doing tension dependent or force dependent measurements is actually a pain point in single molecule biophysics. So that's what we are trying to avoid, or we are trying to solve. And that's basically what Maestro is. But for completeness, I'm going to show you 2 out of many clever tools that people built in the past 25 years. The first one is atomic force microscopy or microscope. It is basically a calibrated needle, and the bending of the needle will induce tension along tether biomolecules. The local expert is in the room, Robert Ross, and other, the pioneers in the fields are Julio Fernandez at Columbia, Hermann Gaup at Ludwig Maximilian University, Dan Fletcher perhaps at Berkeley. The second one is, the name is optical tweezer. The basic theory is that light carries a momentum, and then beats refract lights. And if light gets refracted, there is a change of momentum. As a result, there is restoring force that bring the beats to the center of the tightly focused beam. If we do the calculation, the energetics for a small displacement is basically parabolic or quadratic, basically. So in a sense, we have a linear spring, calibrated linear spring that we can use to apply forces to biomolecules. So all of these machines are amazing, beautiful. It's really fun to build the machines, spending a lot of time in machine shop. But running the experiments with this machine actually is kind of painful because they are slow. They are also, we can only apply forces or a force along single axis. They're also big. But one may argue that the big nature of these machines is the strength, because for big machines like FM, atomic force microscopy, and optical tweezer, we can inject, or we can program, or we can include a lot of intelligence in it, compared to, let's say, if our machine is the size of a molecule. You cannot program a lot of intelligence in molecules. But the problem with the size of these machines is that they are not compatible with many existing tools in many labs. So for example, if Pauline is not here, but if Pauline wants to study force-dependent mechanism using cryo-electron microscopy, we cannot put Avogadro number of FM on carbon grids or Avogadro number of optical tweezers. Therefore, we need a molecular tools to that can grab proteins of interest and apply tension and sprinkle it on the carbon grids, for example, or other equipments. I don't know, fluorometer, gel electrophoresis, anything. All right, so I'm going to show you Maestro. It stands for multi-axial entropic spring tweezer along round shape, along ring shape origami or round origami. Notice the word tweezer. In order for biologists to use our approach, we have to use the word tweezer, unfortunately. So when we design a Maestro, we learn from existing, very successful tension-inducing tools like AFM and optical tweezer. So we came up with a few design principles, and I'm listing a few of them here. One of them is solid or stable support. The other one is calibrated springs. And another design principles is it requires tether, which involve bioconjugation. So I'm not going through all of them in this presentation, but I will touch the support and the calibrated spring. Okay. And in order to apply defined tension or multi-axial tension, we need to also have positional control. So remember that a force have both directions and magnitude. So if we want to apply multiple forces at defined directions and strength, we also need to control the position of the springs, the calibrated springs. Okay. So this is the device. It is a ring-shaped DNA origami. The size is about 45 nanometer, and we will add entropic springs along the frame. Okay. So this is the outline for the remaining of the talk. First, I will briefly introduce DNA origami nanotechnology, and then I will show that we can pattern entropic springs or calibrated springs at least 10 nanometer resolution. And then I will describe entropic elasticity, and then I will show you our data in monitoring fluctuation of model system, namely DNA holiday junction under defined tension and multi-axial tensions. And I will close with a discussion about the current focus and outlook of this work. Good. Okay. So first, I have to introduce DNA origami. So the reason why we use origami is because it's such a wonderful material. It is stable. We can encode a lot of intelligence in it. It's nature's information carrier molecule. Not to prove, but to demonstrate the stability, you can go to many journals and read papers on DNA sequencing of genome from fossils. So that's how stable DNA is. Even after thousands of years, DNA is very stable. For DNA origami is a way to build a nanometer scale structure at arbitrary shape. So the way we do it is we first steal along DNA strand here. Sorry. Well, the long DNA strand is in gray. Typically, we steal it from bacteriophage, and we know the sequence. What we do next is we design short DNAs. They are in color, non-gray strands. And because we know the sequence, we can design the short strands to bring multiple domains of long DNA strand. And then it can fold the long DNA strand into desired shape. Okay. So and then for the short staple strands, what we do is we chemically synthesize them, so we don't rely on bacteria. So we can just change the sequence of the short staple strands. But practically, what we do in the lab is we mix DNA, scaffold DNA, staple DNA in water, add salt, bring it close to boiling, and then let it cool down and close our eyes. And lo and behold, we get DNA nanostructures, DNA origami nanostructures. One thing that is special about DNA nanostructures is that the yield is for a simple structure such as this 2D smiley face. The yield, some papers argue, is 98, better than 98%. For complex structures such as Maestro, the yield is less. Okay. So Maestro, as I said earlier, we chose a barrel-shaped frame. So the reason why we chose a barrel-shaped frame, because we want a stable platform. We want the platform to not fluctuate much because of thermal fluctuation. So the diameter is, the inner diameter is 45 nanometer. And the thickness of Maestro is about 14 nanometer. And if you're not familiar with protein biochemistry, typical size of proteins is about 5 nanometer. Okay. And then what Bima did is he designed Maestro and evaluate image the self-assembled Maestro, and it forms really well. So some of you may think that, some of you may focus on the uncircularized Maestro or dimeric Maestro, but that thing actually can be easily purified. So there are many standard strategies for purification of DNA. I'm showing this slide just because I love my physics 472 students. This is their final class project that they published in PLOS One. And it's interesting enough. So a New Scientist magazine like interview us. So so what they did is they used Lego device to build a gradient of sucrose. And then they put mice, well, in this case, DNA nanotube dimers. And then they use ultracentrifugation. After 3 hours, the monomer travels slower than the dimer. And the top layer is the other impurities. And when they spun or centrifuge for 6 hours, the separation increased. And this is the paper. It was published in PLOS One. So I've shown you the Maestro and Maestro form really well. Next, what I'm going to show you is that we can pattern calibrated springs for applying tension to biomolecules at high resolution. So I showed you this movie earlier. This movie was done by Sean Douglas in probably 2007. It shows the routing of scaffold strands, so strands in gray. And the sequences, we know the sequences, but one thing that is important is that the sequences of each domain, what I mean by domain is this segment here, they are unique. So it's addressable. We know the sequences of each domain, so we can extend the staple strands and use it to place any moieties. You know, it could be gold nanoparticles, entropic springs, fluorophores. So it's basically molecular scale breadboard. That's why we chose DNA origami. So we know that we could place moieties or entropic springs at 10 nanometer resolution, but we tested anyway. This is a super resolution image that my lab took of DNA chip with 12 or 3x4 entropic springs separated by 20 nanometer. And you can see that. And if you're not familiar with super resolution microscopy, is that, or why we use super resolution microscopy, the spacing is 20 nanometer, which is much smaller than the wavelength of light. That's why we cannot just use light microscopy. Okay. And this work is in collaboration with Peter Solk, Hao Yan, which is not in the room, he's picking up his son, Bima, and many undergrads and graduate students. We then push the patterning to better resolution than 20 nanometer. I'm showing you just representative DNA origami chip. The one in the middle is one entropic spring every 50 nanometer. And the one on the right here is a pattern of entropic springs at 10 nanometer spacing, with 10 nanometer spacing. And what you can see here is that for complete disclosure, there is a considerable nuance. Roughly 1 out of 10 entropic springs is missing. So and we don't know whether that thing is due to the synthesis or purification or imaging. but 90% is much better than 0%. So I hope by now I convince you that we have a stable platform that we can purify, that forms well, that we can use to place calibrated springs for the monitoring dynamics of biomolecules under defined tension. Okay. So what is the spring? The spring is, as I said earlier, is entropic spring, and I will tell you what that is soon. Well, I can tell you right now, it's actually DNA. So this is one of the examples that nature is so kind to us. The same type of molecule that we use to build the structure also has mechanical property that we can use as calibrated springs. Okay, and this work is not from my lab. It has been done before. This work was done by Tim Liedel and his student, and Philip Tinnefeld's students. It was published in Science in 2016. Sorry. So what is entropic springs? So to me, I love entropic spring. I think all of you have played with it. It's basically rubber band. Right? So entropic spring for me is probably the most extraordinary concept in physics that I've encountered throughout my journey in physics. So before I came to ASU, I did a lot of work in studying molecular machines, the same machines that cause my lips to open and close so I can deliver this lecture. The name is myosin. So myosin use chemical energy motion. So it's not a surprise that myosin can induce 3 piconewton of force, because all myosins in my heart, in your heart, in my mouth, consume a lot of chemical energy. But rubber band is really interesting because it doesn't consume energy. It uses entropy to generate force, right? So nature tends to go to a state with maximum entropy, with high entropy, or high configurational number of, a state with high configurational number, right? So this is not simulation. This is just 2D self-avoiding random walk trajectories that I generated. So let's say we extend the polymers to longer end-to-end distance. What you see here is that the number of available trajectories is less. And because nature wants to be in a state with higher entropy, there is a restoring force that brings rubber band or entropic spring to a compact state. That's how we will generate force. So it is not a new concept. The pioneers in the field is Doug Smith and Bustamante. They published a few or many science papers in the early 1990s. This is one plot that is based on the equation that they came up with. So basically, this is force extension curve. The main take-home message of this slide is that it's not a linear spring. So linear spring looks like these red lines, and I don't know the right K, so basically it's not linear, right? So it's not Hookean spring. So Robert Hooke famously said many centuries ago that he said ut tensio sic vis, which means as the extension, so the force, that means the force has linear relationship with the extension. So what people say that in this regime, people call this entropic regime. So what we do is not extending the polymer, meaning like we don't deform the polymer like you deform spring. So what we do is we just align both ends of the polymer and let the polymer sample many possible configurations. Okay. Next is I want to show you how we calculate the force. There is another equation from a different paper by Doug Smith and Carlos Bustamante. Basically, if we fix the end-to-end distance and we know the length of the polymer, we can calculate or estimate the induced tension. So in Maestro, we keep the end-to-end distance to 45 nanometer. That is the size of the inner diameter of Maestro. And we can vary the length of the DNA strand. So we can induce, we can change the forces systematically. So right now probably we're switching to a more fun part of the talk, which is the data. Every time we build a new technique, people will ask us to benchmark with like the toy model or the hydrogen atom of biophysics. So what we've been doing is working with Hariadi Junction. So Hariadi Junction looks like this in the middle. It is basically two DNA helices, left and right, that are connected by a crossover points. Okay, and the reason why it actually adopt multiple conformation, but in this conformation, because the red domain stacked with orange domain and green domain stacks with blue domain. Okay. So another thing that I want to let you know that this is not just a model system for biophysics. This is actually relevant to biology. There are many Hariadi junctions in your body right now. It is involved in a process called homologous recombination. It's basically a way nature to increase genetic information in species. So basically, if you have DNA breaks, DNA can bind to another DNA here, and then they can extend, and that way there is a recombination between two DNA helices. But anyway, in our experiments, we benchmark with studying the fluctuations between these two conformations, the one on the left and on the right. So one of the reasons why DNA has a nice staircase structure is because the stacking interaction between nucleotides. The strength of the stacking interaction is actually fairly good, roughly about KT, and the stacking can break. So right now, red domain stacks with orange, but when it breaks, the red domain has two choices, either stack with orange or with green, and goes to the other isomer or conformation, right? So similarly with orange, it can either rebind with red or bind to blue domain. So we have to, it's a nice two-step system, okay? In order to monitor the transition or the fluctuations of Hariadi junction in real time, we conjugate or we stick donor and acceptor fluorophores. So donor and acceptor fluorophores, what we mean by that is we're using FRED or energy transfer between donor molecule, in this case Psi3, and acceptor molecule, in this case Psi5. So the energy transfer depends on the separation between donor and acceptor. So in the state on the left, the FRED efficiency, the energy transfer efficiency is high because Psi3 here is close to Psi5. So DNA helix has a diameter of 2 nanometers. So the efficiency is roughly here, close to 1. For the state on the right, the donor molecule and acceptor molecule are separated by roughly 7.5 nanometer. So the FRED efficiency is low. So by monitoring the up and down of FRED signal, similar to the signal of ion channel, we could monitor the fluctuation of DNA Hariadi junction. In the interest of time, I'm gonna skip all the raw data, all the telegraph signal and how we analyze it. I'm just going to show you the data. But first, I need to explain how we made the measurement. So what we did is we put binder to Maestro that we can use to immobilize Maestro on the cover slip, basically near objective, and we generate evanescent wave by sending the excitation beam at an incident angle that is larger than the critical angle of glass water interface. So basically what we have is a near field close to the surface that only excites Maestro and not anything far away from Maestro. So this is a very common, we didn't come up with this. It's a really common single molecule technique in the field. So the secrets of single molecule experiments are probably only two. One is we have to kill background signal. And second, we need to use expensive camera or a camera with high sensitivity. All right. So for the first experiment, the first question that we asked is, can we use Maestro to immobilize DNA origami without applying tension? So what Bima did is he tethered Hariadi Junction with an entropic spring. And this drawing is to scale, but of course the DNA doesn't look like this. It's just an abstraction. So DNA Hariadi Junction is about 7.5 nanometer, and the diameter is 45 nanometer. So the fluctuation of Hariadi Junction will look like this. It goes from high fret, low fret, high fret, low fret. Okay. So again, I skipped the raw data. This is the distribution of the probability of Hariadi Junction in high fret state and low fret state. The energetics of high fret state and low fret state depends on the sequences near the junction, but zeroth order expectation is that they should be roughly equal. So indeed, what we measure is roughly equal distribution. It's almost 50-50, but we can predict and we can improve our expectation or our prediction. So almost 50% of the time, the Hariadi Junction is in low fret state, and another 50% of the time, or 56% of the time, it's in high fret state. Okay. Next is the fun part, which is, can we apply tension and measure the fluctuations? This measurement actually has been done. It has been done in 2007 by the lab of one of the pioneers of the field. His name is TJ Ha. At the time, he was still at UIUC. He's now at Hopkins. Oh, he just moved. When? Last night? Why he didn't call me? Sorry. Anyway, thank you. So he's at Harvard now. Okay, so basically it was a heroic experiment. It involved confocal single molecule fret setup here and optical tweezer. And people recognize it hasn't been done before, and it's difficult experiments. It made it to science. In some of their measurements, the number of trajectories is 5 because it's difficult to get any data from it. So can we simplify? Can we democratize, this single molecule fret biophysics under tension experiments? So this is what Bima did. He used Maestro, and he tethered a single DNA Hariadi junction on 2 attachment points, and Hariadi junction will perform this fluctuation. Okay, high fret, low fret, high fret, and low fret. And what do we see? Okay, so first, what do we expect? So learning, using analogy with a spin 1/2 particle in magnetic field, we think that this conformation is more dominant because the helix align with the direction of the tension compared to this one, right? This one we expect to be more dominant. So what does the data say? So the one on the left is zero tension, is about 50-50. By applying tension in one direction, in a defined direction, we shift the dynamics from 50-50 to 84% to 16%, 84-16, which match our expectation. And then by increasing the tension, by changing the number of, or the length of the DNA strands, or entropic springs, we further shift the distribution from 84-16 to 97-3, okay? And then what we did is we plot the data. I think it was 46, 84, and 97, right? So those are the data that we have so far. We plot the data on this plot, which is the probability of. I apologize for the unclear symbol. I thought that I was being clever by injecting a picture to LaTeX program. But basically, the probability of low fret is equal to using Boltzmann distribution and precision function, similar to the way we calculated the probability of open ion channels. and then we arrive at this expression. And then the difference of the energy is the amount of work that we put into the system. And then we can plot the model. We can overlay the model with the data. So one thing that I want to point out is that there is no fitting parameter in the model. So we know what F is, which is 3 piconewton, 6 piconewton, 0 piconewton. We know what KT is, and we can estimate delta X from our from basically the geometry of DNA. And in this model, we expect at zero tension, the probability of high Fred and low Fred is 50-50 because I just tried to come up with the simplest model that I can come up with. So we are surprised that the data fits the model really well, perhaps because we generate a lot of trajectories and that way we reduce the uncertainty of our measurements. Or we get lucky, we don't know. Next, the name of the experiment in the lab is Maestro, so multi-axial. So we decided to add one tension, which is perpendicular from the previously used tension. So we connected an entropic spring to basically like this to Maestro. So we have one holiday junction with 3 entropic springs. And the question that Bima asked is, would this configuration can freeze the intermediates? So there is a little bit energy cost here because we lost stacking interactions, but perhaps because the spring does the work, we would be able to see the intermediates. And then based on distance calculation, the intermediate should appear with intermediate Fred efficiency in the middle. This one just to show you that in 2 minutes or maybe 20 seconds, Bima could generate more than 200 trajectories. And you could see that most of them have 2 peaks. But from time to time, you see, for example, the one in the bottom, the distribution of that particular trajectory has 3 peaks. Also here, it has 3 peaks, okay? So by using simple data analysis, we haven't used your fancy non-parametric Bayesian inference. We just collapse all the histograms and create a global histograms. And we get a shoulder between the low Fred efficiency and high Fred efficiency. And we fit it with the sum of 3 Gaussian functions. When we did the same thing with the data with single axis tension, the other Gaussian didn't appear. Basically, we get a third distribution that doesn't make sense. It's not physically meaningful. So I show you data with turf, and then one of the premise of making molecular tool is that the tool is compatible with other machines. So the goal is for scientists to be able to pipette forces. So instead of just pipetting salt, acid base, scientists like physicists can pipette force and use existing instruments in the lab. So one instrument in the lab is confocal single molecule Fred platform. One of the experts on campus, or the expert of FCS in campus, is Marcia. She's not in the room right now. But basically, it's a single tightly focused beam. And Maestro can diffuse in and out. When it's inside the confocal volume for a brief period of time, it will emit light that we can detect. Right? So it's still a single molecule measurements, but we can only measure it for a short period of time instead of like 20 seconds with total internal reflection microscopy. But it's a nice instrument. This is the same machine that my physics 472 used, because you can just put a drop of sample on a dirty cover slip and get a single molecule measurement. Another nice thing with confocal single molecule Fred is that there is no surface sample interactions. Okay. So we, of course, ran the experiments, and this is the preliminary data. We see the same trends as we increase the force. So the top row is data from single molecule turf. The bottom row is data from confocal single molecule Freds. we see the same trend, but not the absolute values of the distribution. So it's a puzzle for us, perhaps because of the surface sample interactions, or perhaps it is just because the low number of trajectories, low number of experiments. So the data on top was taken in, I don't know, 20 seconds. The data in the bottom, it took BIMA more than an hour to construct the histogram. This is because we have to use low concentration, so we don't have, so the probability of having two maestros in one confocal volume is basically zero. Sorry. I don't know. Okay. Sometimes I get too excited and start throwing remotes. Okay, so next, so basically the conclusion of this section is that the model, the dynamics of DNA holiday junction and retention can be tuned by tension, and simple statistical mechanics can describe the dynamics really well. So finally, I want to show you two things. The current focus of the lab. One is to increase the throughput further. So remember, TJH, his trajectories number are on the order of 10 or 5. BIMA could get 200 trajectories in 20 seconds or 2 minutes. Can we increase it to 10,000 or 50,000 trajectories in 2 minutes? So one way to our approach is by creating landing spots for Maestro, and do measurements on those landing spots. So to do that, we use self-assembling colloids or nanospheres. These nanospheres, if you deposit it carefully and you control the evaporation and the condition temperature and solvent, you will generate an order structures. And we use it to as a mask to passivate glass surface. And when we leave the beads, we should end up with sticky patches for Maestros. Okay. So just brief, like physics. I love this theory. This is theory. The name of the theory is Hertzian contact, or Hertzian contact mechanics. As we know, Heinrich Hertz was a theorist, but he also did experiments. Actually, he showed that a light is an electromagnetic wave roughly 3 decades after Maxwell came up with his equations. So in this case, what he did is like modeling, let's say you poke a jello. So you deform a jello. So what this amount to in terms of mathematically is solving green function in half space. And he ended up with a nice expression of the contact diameter as a function of nanospheres that we test systematically. In the interest of time, I'm going to skip that data. But basically, we are successful in creating the landing strips for DNA origami or Maestro. One thing that I skipped also is that there was a different approach by my collaborators, Paul Rothman and Ashwin Gopinath, but they had to pay, but the cost of the experiment is more than $1,000 per chip. So using beads, we could lower the cost because beads is not that expensive, less than $1 per chip. So this is our way to advance science by democratizing nanoarrays, so more people can do more science with like Maestro, for example. But we rely on self-assembly. So we see defects, for example, some spots, they have more than one. So this one is a doublet, but perhaps with filtering, we would be able to exclude some spots, okay? I get it, 10 minutes, okay. So we haven't done experiments with Maestro. This is just to show you the type of data that we could generate, but this is a different single molecule experiments. So each node can be used to recruit a single Maestro. And with that, we can increase the throughput even further. So I only have one more thing, just to close in terms of what we can do. There are many things that we have in mind, and I'm happy to share it with you after the talk or anytime. This is what we wrote to NIH. So we are excited with Maestro, and we wrote a grant, and we received a good score. We received a perfect score. So basically, the reason why they are excited, and we are very excited also with Maestro, is that many biological functions are fine-tuned or depend on mechanical forces. One classic example is this molecule. The name of the molecule is integrins. It gets activated by tension. Okay. So tension is important in biology, but if you look at the number of protein structures in protein data bank, almost all of them were solved without tension. So there is a big need for a technology that can solve protein structures under tension. And this is in collaboration with Pauline and Shu Wang. So the plan is we want to use our Maestro or these molecular robots to grab integrin, apply tension, and sprinkle it on carbon grids and solve the protein structures. So that's one of the direction that we're currently pursuing right now. People have solved the structure of integrin, but in the absence of tension. For example, this is a recent paper by Schumacher and his co-worker in Science Advances in 2021. Okay, so I would like to close, if you don't mind, I would like to close with another passage from the book, the same book, which is What is Life by Erwin Schrödinger. Okay, so let me make it bigger. So let me read it for you. So living matter, while not eluding the laws of physics as established up to date, is likely to involve other laws of physics hitherto unknown, which, however, once they have been revealed, will form just as integral part of the science as the former. The way I interpret this passage is that it's a two-part passage. The first one is that biomolecules, not surprisingly, follow rules of physics, right, as exemplified by the dynamics of Hariadi junction, can be well described by statistical mechanics. The second one is perhaps it's Schrödinger's invitation that interesting science are within our reach, and perhaps new laws of physics. If we are fortunate, we will uncover new laws of physics. So everyone, let's combine forces. Anyway, so the conclusion, I hope you can see how excited I am with Maestro. This is the brainchild of Bima. So Bima is the maestro of Maestro, but I'm excited that he flourished in the lab. So basically, Maestro is a technology that can grab biomolecules and apply defined tension along multiple axes. And we design Maestro we can design Maestro and build Maestro and do physics experiments with it, thanks to many important contributions to the field, such as the invention of DNA origami, the development of entropic elasticity by Eric Sigia, Carlos Bustamante, Doug Smith. And we hope that in the future or near future, Maestro can complement or perhaps rival optical tweezers and magnetic tweezers and AFM. At least it has the word tweezer in it. What is the fundamental limit of Maestro? We have to be fair, right? I think the molecular nature of Maestro is a strength, but also weakness. Because again, it's really difficult to program intelligence into small objects. But I think because of the throughput is very high, perhaps we can suppress the uncertainties of our experiments, as demonstrated by the nicely fit or the good agreement between data with Maestro and simple two-state statistical mechanics model. And the molecular nature of Maestro is nice because we hope that right now scientists can just pipette forces and use existing equipments in their lab to study force-dependent dynamics and processes. Before I finish, I want to thank the real heroes of my lab. My lab is fairly small right now. We have one physics graduate student and one rotation student. The physics graduate student is Bima and rotation student is Josh. We have two very talented postdocs. We have 5 undergraduate students. Three of them are members of undergraduate team that will go to Jimbore in Tokyo in a few months. I'm really excited with their project. We have gap year student, Kavi, and incoming high school student from SU scene. We're working with literally award-winning comic illustrator, Sapto. For collaborator, the work that I show you here is in collaboration. The nano arrays is in collaboration with Paul Rothman and Ashwin. The names are not there because we are no longer working together, but I should have put their names there. The 20 nanometer grid is in collaboration with Peter Sok and Hao Yan. We're really proud of our lab alumni. They are doing really well. It's really gratifying to take them to reach their goals. This one just postdocs and graduate students. I don't have space for undergraduate students, but just briefly, we mentored 3 Goldwater scholars in our history. So I'm really proud of them. For our high school students, we mentor 1 US presidential scholar, 1 Getz fellow, and 1 finalist of Goldwater scholar, if that thing is a thing. It was my daughter. Finally, for funding, we are very fortunate to receive generous funding from NIH Directors, New Innovator Award, National Cancer Institute, also National Science Foundation, Semicin Bio 2 and 3. We receive educational grant. The PI of the grant is not me, it's Abhishek Singharoy. And the nano array project was supported by ABRC, Arizona Biomedical Research Consortium, and BIMA received nice fellowship from American Heart Association. We are doing multiple routes. We are also doing science in an ASU spin-off company. The name of the company is Exodyme. I'm one of the co-founders of Exodyme with Hao Yan. We received generous funding from a venture firm from San Francisco and from Intelligence Advanced Research Project Activity, or IARPA. With that, thank you for your attention.